Hello students, how are you? This is 11 Biology Practical. Today we are studying all these three activities. Okay? The main experiment is to study the effect of enzymes on starch, egg albumin and fats. Okay? In first activity, aim is to study effect of salivary amylase on starch. For this requirement is test tube, as usual test tube stand, then salivary amylase, starch, distilled water, etc. Okay. In procedure, we take three test tubes, create setup of three test tube, label them 1, 2 and 3. Okay. After that, there are common ingredients in each test tube. Okay, that common ingredients are 5 ml starch plus 3 ml of buffer. Add these two ingredients in three test tubes. Then the next ingredient is change in each test tube. Add with that 3 ml of salivary amylase in test tube number 1 only. Okay. Then in test tube number 2, with this ingredient add 3 ml of distilled water only. This is for test tube number 2. For test tube number 3, with this ingredient, common ingredient, add 3 ml of distilled water and 10 drop of dilute HCl. Okay. This is for test tube number 3 and this is setup of 3 test tubes. Okay. After this, there is incubation process of 3 test tube. Incubate these 3 test tube for 15 minutes at optimal pH 6.7 and at 37 degrees centigrade. Okay. This process is for 10 to 15 minutes. Then remove the test tubes and add Lugol's iodine solution. Lugol's iodine solution is an indicator for test of starch present in test tube. Okay. So after adding Lugol iodine solution, we get results in 3 test tubes. There is color change. In first test tube, amber color is seen. So protein is digested. Okay. Sorry, starch is digested. Here the test is for starch. In test tube number 2, we see black color. So starch is undigested. In test tube number 3, color is blue black. The color is blue black. So the protein remain undigested. These are 3 different results. So write in your practical these observations separately as conclusion and in also observation table. Okay. Then activity number 2, effect of pepsin on egg white or egg albumin. This is second activity coming under this practical number 10. Okay. Egg white or egg albumin is extracted from egg. Egg containing two type of egg, egg white and egg yellow part. So extract only egg white for this practical. Okay, collect them into the beaker. So principle for this activity means principle related to enzyme pepsin. Why pepsin? We are using enzyme pepsin on egg white, and we are going to see the results. Okay, pepsin is a protease enzyme. It used for digestion of protein. Okay, it break down proteins into peptide bonds. So this pepsin, how pepsin we get in the stomach? Pepsin is generated in stomach by pepsinogen. See here, pepsinogen. From pepsinogen with HCl, we get pepsin. Okay, pepsin is a enzyme which is used for breaking of proteins into peptide bonds. This is the main principle for activity of effect of pepsin on egg white. Okay, after this we are taking procedure. In procedure, create setup of three test tubes, test tube 1, test tube 2 and test tube 3. Label it 1, 2 and 3. Okay. Then the common material we are using in three test tubes. Do you know what is it? It is 3 ml of egg white. Add 3 ml of egg white in each test tube. In each 1, 2, 3 test tube. Okay. This is common ingredient. Apart from this, in test tube number 1, add 5 ml of dilute HCl. Okay. This is for test tube number 1. For test tube number 2, apart from adding egg white, add 3 ml of pepsin plus 2 ml of dilute HCl. Okay. This preparation is for test tube number 2. And for test tube number 3, we already added 3 ml of egg white and then add 3 ml of pepsin and 2 ml of NaOH. Okay. After this, creating the setup, we incubate these test tubes in 37 degrees centigrade, and this incubation process is for 10 to 15 minutes. After that, we get result. So write down your results and observation in observation table. In conclusion, you have to write. We see in first test tube solution is milky. Okay. This indicates no digestion of proteins. In test tube number 2, solution is clear but there is digestion of protein. Okay, pepsin breaks down protein into peptide bonds, so the solution is clear and there is digestion of proteins. Okay, in test tube number 3, what is conclusion? The solution is clear, but there is no digestion of protein. Okay, why? We added here 2 ml of NaOH. See here, we added NaOH. So, effect of acid is not there. Effect of acid denatured by NaOH solution. Okay, so we are not getting result of digestion here. There is no digestion. This is about whole about the effect of pepsin on egg white. Okay, now we are taking activity 3, emulsification of fat by bile juice. Okay, so aim of this experiment is to study the action of bile on fat. Okay, this is our aim of experiment, and requirements for this procedure as usual: test tube stand, bile juice, distilled water, dilute HCl, NaOH, etc. So, in procedure, create, sorry, we have not taken principle for this. I am taking this. Principle for this practical is bile. See, the bile juice and pancreatic juice, they are forming our stomach. Bile juice produced in our liver and stored in gallbladder. First, when we eat the pre-digested food, it goes to mouth, then our stomach, then in intestine. So, bile juice is secreted by liver whenever it requires more in intestine. It is secreted by when the food is in intestine. Okay, bile juice is stored in gallbladder. Bile helps in breaking liquid in tiny droplets. So, what happen? Undigested food, they are break down into tiny droplets, and it prevents getting two separate layers of that liquid. So this is the process or technique of emulsification, which is it is useful in this experiment. Okay, so student, you know, emulsification, it is a process of breaking down liquid into tiny droplets. It prevents two separate layers of water and that liquid. Okay, so emulsification is necessary in our digestion of food. Okay, this is the principle for this activity of emulsification. In procedure, we are taking two test tube. Create a setup of two test tube, number 1 and number 2. Label it. Then in each test tube, add 3 ml of oil in each test tube. Okay. Then add 3 ml of distilled water in test tube number 1. Okay, only add 3 ml of distilled water. So the ingredients in test tube number 1 is 3 ml of oil and 3 ml of distilled water. You got it? In test tube number 2, we have already added 3 ml of oil. Okay, then add 3 ml of bile juice. Okay, so after it, shake it well, the test tubes, and see the results and observation. Write your observation in observation table. Okay. In conclusion, what we get in test tube number 1, there is no emulsification of oil. Okay. And in test tube number 2, emulsified fat is observed. In test tube number 1, we seen there are two separate layers. One layer is of oil and one layer is of water. So, oil is not emulsified. Okay, it is not getting, uh, there is no process of emulsification. There is no tiny droplets in test tube number 1. But in test tube number 2, the oil is emulsified. Okay, we find small tiny droplets of oil that is suspended in water. Okay, this is whole practical of practical number 10, 11 biology. Okay, so you have to write observation table, color changes and conclusion in these 3 different activities. Okay, so student, if you like the video, you check out other published videos and subscribe our channel and write your comment in comment box.